So let's begin with another session for today on QBD in formulation development. We have a distinguished speaker for today's session, Mrs. Meeta Jain ma'am. Mrs. Meeta Jain ma'am is currently working as an assistant professor in pharmaceutics department at Central Institute of Pharmacy and Research. She graduated in pharmacy from the University of Mumbai in the year 2010, subsequently obtained a master's degree in pharmaceutics from NIMS University, Mumbai in the year 2012. Currently, she is pursuing her PhD in pharmaceutics from the University of Mumbai. She has 10 years of teaching experience in the areas of industrial pharmacy and drug regulatory affairs. She has received minor research grants from the University of Mumbai and has published articles in various national and international journals. With this brief introduction, now I would request ma'am to take over the session.
Good morning, everyone. Thank you for the kind introduction. I'll just share my presentation. Okay, good morning, everyone. So I'll be taking you through a session on quality by design in formulation development. Okay, so basically, if you have with you the API, the drug, and excipients, how we are going to develop it into a dosage form, that is a formulation, so that it can be helpful for the patients to diagnose their disease, to cure or to prevent. So basically how to apply this quality by design, which is now a essential part of formulation development throughout the world. The regulatory agencies require all industries and researchers to apply this in formulating their dosage forms. So before I go into the topic, I'll take you through the traditional approach which was used around 15 to 20 years back. Okay, so if there is a path where you have to reach to a destination B from a starting point A, initially they used traditional approach, which was just focused on achieving compliance to product specifications. Like they have to reach to B anyhow, whatever they take, right, left, they go up, they go down, they have to reach to the position B having a specified product specifications. Only minor variations were studied to establish that the product is reproducible. So there were errors when others tried to go from that path from A to B or try to prepare that particular product. Okay, it was not reproducible, maybe because there were variations which were not studied. Then it was not necessarily the best way to get from A to B. So what is your QBD approach is they just don't have the aim or goal to reach the destination. They try to find out what is the best way to get from A to B. What are the other routes can I take to safely get from A to B? So it's basically like a GPS navigation system. You have your Google Maps, which give you options of various paths from where you can reach from A to B. What is the best path, keeping in mind the time required, the traffic which is there from moving from A to B. So similar is our QBD, which is a GPS navigation system for product and process development. So before that, What is quality basically is the suitability of either drug substance or drug product for its intended use. This term includes attributes such as identity, strength, and purity. So if your product has proper a drug which you require it out of appropriate strength and purity, that means it is of a desired quality. And quality by design is a systematic approach to formulation development that begins with predetermined objectives and emphasizes product and process understanding and process control based on sound science and quality risk management. So basically, the quality by design uses a sound scientific approach. It's not just like you are taking something and manufacturing a product of desired quality. You have to apply your scientific knowledge, okay? And there are predetermined objectives which you have to keep in your mind. That is, what is the final product which you require, what quality it should have. That has to be predetermined, okay? And it also has a huge understanding, like a huge process of understanding what is your process, what excipients you are going to use in that, and how you're going to control the quality of your process. So this quality by design is a concept first developed by a quality pioneer, Dr. Joseph M. Juran. He believed that quality should be designed into a product and that most quality crises and problems relate to the way in which product was designed in the first place. So if you are not starting with the proper quality or proper aim or proper things, you might have quality issues at the end. So he has saying that quality cannot be tested into the product. It has to be built in by design at the first. So basically, quality by design uses a science and risk-based approach. So it studies how your excipients and the process are having risk on your quality control parameters. Okay, and that emphasizes importance of developing scientific knowledge and thorough understanding of both product and the process. So a successful QBD approach leads to systematic development process with better understanding of products. So you have scientific knowledge. Okay, you need to have process understanding. So then you are going to design your product and control your product. That is a whole of your quality by design. It leads to more robust manufacturing processes, and it has potential for timely and flexible regulatory approval because you are designing. So there is some space in which you can move in that particular design so that you will have the same quality end product by moving in that particular design. So that is why you have a flexible regulatory approvals. And according to Food and Drug Administration, QBD means designing and developing a product and associated manufacturing processes that will be used during product development to ensure that the product consistently attains a predefined quality at the end of the manufacturing process. Okay, this is very important. The quality which has to be there in your final product, okay, and the quality how it should be, like what we have in mind at the start. So it is a predefined quality, and it also ensures a robust process. That means it ensures that every time you go through this process which you have developed using quality by design, you will get, and there is a surety that you will get consistently the product of the same quality which you always get. So initially it was like hit and trial or trial and error methods which were used. So now you're moving away from that hit and trial method and using QBD, that is quality is built into product not only by controlling by testing, but you are building the quality in that product. The foundation for pharmaceutical companies to implement QBD in their operations and product development has been laid down by several ICH conferences, ICH guidelines, that is international conferences on harmonization guidance documents. That is ICH Q8, which is basically gives you guidance on pharmaceutical development. ICH Q9, which gives you the information or guidance on quality risk management. ICH Q10 guideline, that is quality 10 guidelines, gives you pharmaceutical quality system. So thereby, if you go and go through this guidance, these are just the guidance. It does not give you any method for QBD. It is just a guidance. You will get the product of a good quality, and that is also a life cycle approach. Till your product is there in the market, you can apply this quality by design. Whereas another guidance, that is process analytical technology, that is one of the tool of quality by design that is provided by FD. So one of the advantages of QBD is, first is better understanding of the process, and so less batch failures, then more efficient and effective control of change. So there is a design space which is developed, which I'll take you through how you develop a design space. So if you are forced to change something in your formulation, like material or process, due to some unavoidable circumstances, you can move in that space. So there is more efficient and effective control of change. Then return on investment and cost savings because of less batch failure and more effective change control. Also, there is additional opportunities for more flexible regulatory approaches. Okay, that is, for example, manufacturing changes within the approved design space without further regulatory review. So if you ensure regulatory agencies that you are moving only in that design space which is created by QBD, then they won't require much review. So what is design space is like a space in your classroom where only you can sit for your lectures. So wherever you sit, you will be able to get the same thing which the professors are teaching you. That is an example of design space. Okay, so wherever you will move with your variables in your formulation, you will get the same results if you move within that design space. Then it is a risk-based approach and identification. So you know what are the risks. So for the development till the end, you will know what are the risks associated with that process. So you will take care of that. Then reduction of post-approval submissions. So as I told you, if you move within the design space, then you won't require much submissions. Then better innovation due to ability to improve process without resubmission to FDA when remaining in the design space. And lastly, more efficient technology transfer to manufacturing, that is from lab scale to commercialization. It becomes easier to transfer your technology which is developed in the lab if you are applying a proper quality by design. So next part is the elements of quality by design, which are very important, and these are also, you can take it as a steps how you have to go about quality by design. Okay, so first is a target profile. Okay, so first is a target profile. So as I have told you, the quality by design, you have to have a predetermined objective. So this is the target. So in beginning itself, you have to have a target in your mind so that it will be easier for you to reach to that target. That is called as quality target product profile, that is QTPP. Okay. Then from this, we come to what are the critical quality attributes. So what is your target, and of those targets, what are the critical quality attributes which can be affected by your various excipients which you are using and by various process you are using for manufacturing that formulation. Then next is risk assessment. Okay, so basically risk assessment is one of the tools, but in this, the two major elements of QBD, that is material attributes and process parameters. how you're going to link them to your CQAs and perform risk assessment. Okay, so after that, after deciding what are the critical quality attributes, you have to decide what are the raw material attributes which can affect that critical quality attributes, what are the process parameters which can affect that critical quality attributes, and then perform risk assessment. That is how potential risk they have on those critical quality attributes. After you determine the risk, wherever there is a high risk involved, you have to evaluate it using design of experiments. You have to create a design space and use design of experiments to evaluate that risk. Then you have to establish a control strategy and strategy for continual improvement. Okay, so these are the elements. Basically, after designing, you go for scale control strategy, then scale up, exhibit batches, submit it to regulatory agencies. Post approval, you require this step, which is a continual improvement till your product is there in the market. That is still the product life cycle. Okay, so to perform or to use these elements, there are various tools which are there to facilitate this QBD implementation. That is prior knowledge, by which you derive at the target profile and CQAs, then risk assessment, then design of experiments, and process analytical technology. So we'll basically go through all these elements in this session. First is quality target product profile. So it is a prospective summary of quality characteristics of a drug product that ideally will be achieved to ensure the desired quality, taking into account safety and efficacy of drug products. So it is a prospective summary type, okay, of the quality characteristics, which looks into consideration the safety and efficacy of your drug product. So to ensure that your drug product is safe and efficient, what quality or what characteristics you require in your drug product, that summary usually is defined in QTPP. So it is a basis for formulation design and is derived from understanding of the mode of action of the product, patient profile, clinical indication, desired safety profile, and quality characteristics which are related to route of administration, intended use, dosage form, then which delivery system, what is the dose strength, even container closure system, then drug product, dosage form which is being developed, then the parameters which are going to affect your pharmacokinetics characteristics, that is, for example, like dissolution. Then what is the product quality you require, the criteria like sterility, purity, stability, and drug release, which are appropriate for the marketing that product. Okay, so next slide. Okay, and the QTPP is different for your generic formulations and innovators formulations. So your QBD, or you can apply your QBD, and you have to apply this to both generic as well as innovative formulations that is required by regulatory agencies. So even if you are developing generic, you require to apply this QBD process. Okay, so for generic formulations, the target is defined early in development, and it is basically based on properties of your drug substance, characterization of innovator products. So generic formulations basically is you are going to make a similar product like innovator, which is also that is why it is called as reference listed drug. So innovator product is called as reference listed drug, which you are going to refer to make into your generic formulation. So in consideration of the RLD label, that means what dose they have used, that is how much amount of drug substance or API they have added in your formulation, you need to keep it same. Okay, and what is your patient population which you are targeting. So it includes all product attributes that are intended to ensure equivalent safety and efficacy to the innovator's product. In case of new formulations, which are innovative formulations, the target is based on the research which you're going carrying out, and desired properties of dosage form you require in your new formulation. Okay, so this is basically the quality target product profile for generic immediate release tablets. Okay, so if you can see the elements are dosage form, which dosage form. So in case of generic, you just, and very important is you have to give justification. The regulatory agencies require this, and justification that why you are having that particular target and why this is a element of your QTPP. Okay, so the dosage forms like tablets, why? Because it is a pharmaceutical equivalent requirement, that is a same dosage form. So in form, while formulating generic, you require it to have a same dosage form as innovators. Similarly, the dose design has to be similar, that is immediate release, so that it will meet the label claims. Then the route of administration has to be same as the innovator, so it is oral, and they have given that particular justification. Dose strength has to be equivalent. Okay, and pharmacokinetics, you have to prove that it is bioequivalent to your innovator formulation. Okay, so bioequivalent to your innovator, that is also called as reference listed drug. Okay, and stability, same, at least 24 months shelf life at room temperature, equivalent to or better than your reference listed drug. Okay, then drug product quality attributes like physical attributes, identification, assay, content uniformity, dissolution, degradation products, residual solvents, water content, microbial limits. These are all the quality attributes which your drug product should have. Those also are pharmaceutical equivalent requirements in case of your generic formulations. Okay, then you can design your own container closure system, your own labeling, but it has to have the label has to have the similar kind of information which is there with respect to the dose strength and the amount of API. Okay, and whatever container closure system required to achieve integrity during shipping and maintain the shelf life. Okay, and there are no alternative methods of administration because nothing are listed in R&D label. So this is the quality target product profile for generic. Now we'll see how it is different for your parent novel formulation. So basically this is a novel parental formulation of microparticles. Okay, so there is no reference listed drug, so dosage form you have decided it has to be a microparticles because of the drug properties. Might be a hydrophobic drug, so reduce the size, which can can be injected intramuscularly or subcutaneous after reconstitution. Dosage is parenteral, so you can see the justification. I just want to differentiate between the QTPP for novel formulation and generic, and see the justification there. The justification was just it has to be pharmaceutical equivalent, whereas here you have to justify it as per the research which you have carried out and what properties your dosage form should have at the end. So like release properties, okay, and how you're going to inject it, which type of action you require, the frequency. Okay, then the drug product quality attributes, these are the parental, so it will differ. Like particle size is important, and encapsulation efficiency is important, then potency, impurities, residual solvents. Then similar to that you have in vitro drug release profile. Then another important thing is sterility, endotoxin test, pyrogen test, because these are parenterals. Another difference is in the different types of dosage form which you want to prepare. So in case of tablets, your drug product quality attributes were different. In case of parenterals, your drug product quality attributes are different. Okay, and you have to give proper justification. And here you don't have any reference drugs, so you need to give justification as you want like how it affects the efficacy, how it will affect the stability or quality of the product. Okay. Then like stability is there. Then what is the yield that is important in case of this microparticles, okay, and container closure system. Next is critical quality attributes. So critical quality attribute, that is also called a CQA, is a physical, chemical, biological or microbiological property or characteristics that should be within an appropriate limit, range or distribution to ensure the desired product quality. Okay. So it includes all physical, chemical, biological, microbiological properties. Okay, and it should be within a limit so that you will get a desired product quality. Okay, this CQAs, how you derive the CQAs, these are derived from quality target product profile and is based on severity of harm to a patient that should the product fall outside the acceptable range for that attribute. So suppose for one of the attributes, your formulation or product goes outside the acceptable range, how severe harm it will have on the patients. That decides whether it can be a critical quality attribute or not. So for example, if you can see in this picture, the dissolution, dose, appearance, hardness. These are the attributes of a tablets. How severe severity it affects, or how severely it affects the patients if it goes out of the acceptable range. For example, resolution, if it goes outside the acceptable range, it will have a severe problems in the patient because the drug will not dissolve as desired, so it will not be absorbed and will not show the correct action. Whereas in case of appearance, you have a target of appearance that it should be smooth and uniform in color. But if it is not so, okay, it will not have a severe impact on the health of the patient. So it might not be considered as a critical quality attribute. So identify what is critical to the patient and link this to the drug product. So how I will identify the CQAs? They are generally linked with drug substance, excipients, intermediates, that is in-process materials, and drug product, as well as to the process. Okay. For example, CQAs of solid dosage, solid oral dosage forms are typically those aspects affecting product purity, strength, drug release, stability, whereas for parenterals, they are sterility and clarity. Along with that, additional properties like particle size, bulk density will also affect the these dosage forms, the release or the quality of this dosage forms. So mostly derived from quality target product profile or with prior knowledge, which can be used as a guide, is used to guide the product and process development, and subsequently CQAs are assessed for risk management. So that we'll see in the next slides. So you have a target in your mind. Okay. Now you have to decide what is the what potential impact it has to the safety, efficacy, and quality of your product. So if it has no potential impact, then it will not be considered as a CQA. If it has the potential impact, what is the severity? You have to decide or find out what is the severity. So it's all done by prior knowledge and literature. What is the severity of that target on the patients? Okay, how severely it can affect. If it has low risk, if it has high risk, okay, it is considered as critical. So you have to evaluate that, bring that risk to a low risk, and then proceed with the final formulation. Okay? And continuous improvement is always there while you are lowering the risk in case of parameters which have high risk. So a severity scale is used to assess relative magnitude of impact. A change in criticality only occurs with a change in severity. And what target profiles can be called as CQAs is, there are two types of CQAs. One are potential quality attributes and other are critical quality attributes. So first you'll define the target according to safety and efficacy, then identify the potential quality attributes, that is, and this is in case of your tablets. Okay, then out of these potential quality attributes, what are the critical attributes, okay, which will affect the quality, that is in case terms of potency, purity, safety, identity of your product, and that will be called as your CQAs. Okay, so these are the CQAs for a generic immediate release tablets. Okay, so as if you remember, we have seen this physical attributes and defined it in your QTPP. Okay, what is the target? Is that a CQA or not? Okay, so appearance, color, shape, acceptable to the patient. No visual tablet defects should be observed. That is your target. Is this a CQA? No. Why it is not a CQA? That is also to be justified, that it is not directly linked to the safety and efficacy, so they are not critical. The target is set to ensure patient acceptability. Okay, similarly in case of odor, then similarly in case of size, because it is similar to reference listed drugs, so this is for generic, so it is similar to reference listed drugs, so it is it might be ease, or it has ease of swallowing, or similar acceptance because the reference drug was accepted. Okay. Then scored configuration, that is target is unscored, that is the RLD is an unscored tablet, so therefore generic tablet will be unscored, so it is not critical. Then case of friability, because the routine test you perform and it is as per the compendial requirement, so it will be always less than 1% weight by weight. If it is more than it fails in quality control, so it is not considered as a CQAs. Whereas identification, assay, content uniformity, and resolution, these are the targets, and they are critical because variations in this will cause variations in the how it is going to show effect, that is efficacy and safety of your product. So if identification, okay, but identification you will not take it further in your risk assessment, because this is critical, but you will not be assessing it for risk assessment because it will be monitored during drug release and assay that whether the drug present is actually your API or not. So it will be evaluated further. The parameters like assay, content uniformity, and resolution will be evaluated in further steps, okay, of while applying elements of QBD. Similarly, you have CQAs for novel parenteral. So this all are connected to your QTTP, which we have seen early in the earlier slides. Okay. So in this case, the morphology or appearance is important. Okay. In case of novel parenteral, morphology and appearance is important because you require free-flowing dry powder with limited pores. If pores and cracks are more, that is, if your surface or the appearance of your microparticles are not smooth, then there are chances that there will be a burst release. Okay, that is, all drugs will be released or much of the drug will be released initially, and there will be a lag time later on, which is not desired. So this is a CQA. Similarly, particle size distribution is a critical attribute because this is a parenteral formulation which is directly injected into the blood circulation. So it has to be released from the syringe and also has to circulate in your capillaries. So this is important. Encapsulation efficiency is also important because it will affect the efficacy. That is, if less amount of drug gets encapsulated, it will be an issue. Similarly, potency, impurities, residual solvents, in vitro drug release. Okay, then your sterility test, microbial limits, endotoxin, all these are CQAs for your parenteral formulations. Okay, then even stability, okay, and container closure system is a CQAs. You have to give proper justification. Most of the time, based it is for safe and efficient final drug products. Next is next part is we'll go to the next elements like linking the material attributes and process parameters to your CQAs through risk management. So for that, this first is a step is to identify all possible known material attributes and process parameters that could impact the performance of the process. Okay. So same case study will continue. So before that, what are the critical material attributes and critical process parameters? So critical material attributes basically are physical, chemical, biological or microbiological properties or characteristics of an input material. Okay, that should be within an appropriate limit, range, distribution to ensure desired quality of the output material. So what you are putting into your formulation, okay, that is the what material. So in the common words, these are the excipients as well as API, which you are putting together to get your desired output material, which is nothing but your final formulation. Okay. And a critical process parameter, the second part, which is an measurable input, that is an operating parameter, or output, that is a process state variable of process step that must be controlled to achieve desired product quality and process consistency. So this CMAs are related to your excipients and API, which you're going to put into some of those equipments like process, the unit operations to get the desired product. Those are called as process. Okay. So what steps, what are the measurable parameters in that particular process? So for example, if you are taking a blender, okay, a blender has blender rotation, the RPM, the load which you're loading in that blender, all those are the operating parameters of those blenders, and you have to evaluate whether they are critical or not. So a parameter is critical when a realistic change in that parameter can cause product to fail and to meet the target profile. Okay. So example is taking the same case study of immediate release tablets, of which we have developed the target profile and identified the CQAs. The material which they have used in that, the excipients is lactose monohydrate, microcrystalline cellulose, cross carbonylose sodium, talc, and magnesium stearate. These are your disintegrants. These are the diluents. These are the disintegrants, and these are lubricants and your glidants. Apart from the API, which you are going to which are your input things. Second is the CPPs, that is process parameters. So these are the process steps which are going to be followed to manufacture the tablets, which are compaction, then blending and compaction, blending and pre-compaction, blending and lubrication, then roller compaction with milling, then final blending and lubrication and compression. So this step basically is for the intragranular agents which you are going to add, that is your lactose and MCC and cross carbonylose. Okay, then you are going to compact it and reduce its size, and finally you are going to add talc and magnesium stearate as lubricants, glidants, and then compression. So these are the various operating parameters which you can change for that particular equipment. So you need to study, if you are moving away from this number, how it is going to affect your CQAs. Okay, if you are using more pressure, then how it is going to affect your CQAs. Okay, so you have to map the input material, that is input is material attributes as well as process parameters to your CQAs. Okay, so relationship is, suppose CQA 1 is a function of material attribute 1, that means first material which you are giving a code as material 1, M1 is going to affect your CQA 1, then it has that relationship. CQA 2 is affected by P1, that is one process parameter P1 as well as process parameter P3, then it will be a function of P1 and P3. CQA 3 is a function, or it is going to be affected by M1, M2 and P1, so it will be a function of that. Okay, and P2, none of the CQAs is affected by your P2, so it will not be needed for establishment of the design space. So you don't have to evaluate this for creating your design space. Okay, so basically these are your input, and what is your output is your product. How should be your product? It should have all these quality attributes which you are terming it as a CQAs. Okay. So next step is now you have identified all the possible known material attributes and process parameters that would impact performance of your process. Second is use risk assessment and scientific knowledge to identify potential high risk attributes and parameters and identify levels or ranges of these potentially higher risk attributes and parameters. Okay, so you have to identify the levels of risk which the material attributes and process parameters have on your CQAs, that is critical quality attribute, using a tool which is called as quality risk management. So it is a strategic safety program designed to decrease product risk by using one or more intervention or tools. So you have various tools like fishbone diagram and failure mode success, effect analysis. So these are the various tools which are used for QRM, that is quality risk management. So it is a systematic process for assessment, control, communication, and review of risk. What are risks? Are basically the harm which can be caused to your final product quality by various parameters or the input which you are putting in to get the final product. And it has to be done throughout the life cycle of your product. So two basic primary principles are there. Evaluation of risk to evaluation of the risk to the quality should be based on scientific knowledge and ultimately linked to the protection of the patient, and the level of effort, formality, and documentation of the quality risk management process should be commensurate with the level of risk. That is, if something has a high risk on a particular CQA, you have to put in all the efforts to reduce that risk to a low risk. If it is having a low risk, no need to check out or put more efforts on them, how it will affect your CQA. So what is the severity or the level of risk or the harm that particular parameter is going to cause on your final quality of your final product? That as per that you have to put the efforts. Okay? So this is the same case study again, the initial risk assessment of the formulation variables, that is the materials or the API and excipients which you are putting into producing that particular or developing that particular formulation. So we have seen you have drug substance, that is your API. We have used MCC and lactose as diluents, cross carmellose sodium as disintegrate, talc and magnesium stearate as glidants and lubricants. And we have identified assay, content uniformity, dissolution, and degradation products as your CQA. So these are the critical quality attributes. How severe is the risk of drug substance particle size, on content uniformity, on assay, on dissolution, degradation products that you have to list down. Similarly, of microcrystalline lactose ratio, of cross carmellose sodium level. So this is an disintegrant. So it will have a high impact on dissolution, whereas it might not have much severe impact on assay, content uniformity, and degradation products. So those are termed as a low risk. So you can see here, low risk means broadly acceptable risk, no further investigation is needed, whereas medium is risk is acceptable, further investigation may be needed in order to reduce the risk, but risk is acceptable. And so in this case, if you can by a proper efforts, if you can reduce this, it is good enough, or else it is also acceptable. Whereas where there is a high that is a red color, the risk is unacceptable, so further investigation is needed to reduce the risk. And ultimate goal of this QBD process is all this yellow and red should turn into low, that is your design space and your control strategy. Okay. Similarly, in case of process parameters, now this I am taking only with one of the sorry, these are the way all the unit operations involved, which we have seen in the earlier slides, like pre roller compaction, blending, lubrication, then actual roller compaction, milling, final blending, and then compression. So all these parameters, what are the risks they have on this drug product CQAs, okay, whether they have medium, high, or low risk. So for example, if you see the final blending and lubrication, it will have high impact on dissolution. Why? Because you are adding talc and magnesium stearate in the final blending, and if you are adding more amount of magnesium stearate, so magnesium stearate has a more severe impact. So we have seen here it has a more severe impact than talc. Say, although both are hydrophobic, but the impact of the percentage or level of magnesium stearate which we are adding will have more on dissolution and degradation products. Okay, so here dissolution, it has a high risk. Okay, in case of compression, SA medium, but in case of content uniformity and dissolution, it has high risk because if the proper flow is not there, and the machine is not running if properly, then it can create an issue with content uniformity, and if you're not using a proper pressure, then it can have effect on dissolution. Okay, so this is initial risk assessment on basis of whichever parameters have high risk on CQAs. You have to evaluate them and create a design space using design of experiments. So next step is the design and conduct experiments using design of experiments. I'm not going to go in detail because there was earlier a session on this. So there are various analyze them. So conduct the experiments using design of experiments, and after conducting, you will evaluate your formulations, do the quality control evaluation, and analyze the data which is obtained from that. Okay, and see whether material and process parameter is critical, how critical, and you have to reduce that criticality or severity to low. Okay, so design of experiments is required when you have a multivariant statistical design, that is where many factors are involved, and one factor is having interaction with the other factors which will affect your final CQAs. Okay, so a univariant method, that is one factor at a time, is acceptable in cases where there is no potential interaction between the factors. Since this is not often happening, we require a multivariant statistical design, that is design of experiments, is often used and results are evaluated within commercially available statistical software. So there are various softwares available to perform this design of experiments, where you have to first screen the factors, then build a model, optimize the factors, optimize your formula, perform the test, and measure the output. So you are going to analyze it using various statistical data like ANOVA, okay, and see how potentially your formulation variables interact with each other and how it affects your CQA. So this is same case study where we have developed QTPP CQAs. We have identified critical material attributes and critical process parameters, the risk to CQAs. So we are choosing those formulation variables. So if you remember the table of formulation variables, the drug particle size, disintegrant level, and the ratio having a major impact or having high risk on CQAs, so we'll evaluate them. Even magnesium stearate had a high impact, that is done in a second formulation study. This is first formulation study based on intragranular excipients and drug substance, which you are adding before granulation, whereas magnesium stearate you are adding after granulation. So there is a separate study for that. So 2�3 factorial design is for having the two factors and levels, okay, in which you are going to this will be 3 raised to 2, 3 raised to 3, because 3 factors and 3 levels you are evaluating, and you are going to check the effect of all these variables on this all responses. Okay. Dissolution, disintegration, this is the force by which you are going to perform the compression. Hardness on all these various forces, friability on these various forces, and degradation products. So goal is to maximize and minimize, and these are the acceptable ranges. So after putting this input in your design expert software, you will get the various batches which you have to prepare of these various combinations. You have to prepare those batches, then evaluate them for all these responses, input the result of these responses, and it will give you the whether your design is acceptable or not, and what is the impact on CQAs, that is this responses, and how you can reduce it, and what will be your design space. Similarly, this is for a milling process. Okay, that is one of the process step or unit operation you can call, that is roller impaction and integrated milling process. So roller pressure, roller gap, mill speed, mill screen, orifice, these are the variables which have high impact on the CQA. So you are evaluating that. You are using various levels, means this are the different pressures which you are using, the gap between the rollers, the speed of the millers, and the orifice. So how it will affect all these responses, those are nothing but your CQAs. Okay. Similarly, you have to put this in software. It will give you the combinations in which you can use this, prepare the batches using those process variables, evaluate them for this responses, input in the software, and by performing analyzing using ANOVA, it will give you the results. Those are having a greater interactions with each other and impact on your final quality. So after performing, this is just for one of the unit operations. Similarly, you have to perform for all the unit operations, taking all the process variables. Then after this, after performing on all the process parameters and all material attributes, you will be able to create a design space, that is a multi-dimensional combination and interaction of input variables, that is material attributes and process parameters, that have been demonstrated to provide assurance of quality. So this is actually a depiction of the knowledge space which we had initially, when we had defined the target. After performing, we have come to this confined space that is called as design space. So as I told you earlier, if you move anywhere in this design space, you will get the same output. Okay. And control space is required for having, how you have developed that particular product, your product. Okay. That comes under this control space. So working with a design space is not considered as a change. And last step is to develop a control strategy, that is this control space. Okay. So how you will develop this is, for that, just what is control strategy? It refers to a set of planned controls derived from current product and process understanding that ensures process performance and product. So current product. Okay, so whatever you are producing using a particular process, okay, what should be a particular parameter, how it should be, that is the limits. Okay, so one of the important parts of control strategy is to establish a control system. So it is nothing but defined controls and their established acceptance criteria, that is for limit, based on product understanding that assures product quality. Okay, so establishment of acceptance criteria, that is an important term. What is acceptance criteria? Is the range which is accepted, that is in that range you will get that if that particular parameter is there, you will get the desired quality of your product, which has proper potency, safety, purity, efficacy. Okay. And the control includes parameters and attributes which are related to drug substance, drug product, its components, drug substance, drug product materials, its components, facility and equipment operating conditions, in-process control, finished product specifications, and all associated methods and frequency of monitoring and control. Okay, so same case study. These are the control strategy of material attributes. So we have lactose, microcrystalline cellulose, gross carbonyl sodium, talc, magnesium stearate. Okay, lab scale, what we have done. Okay, after defining design space, you will go for a pilot plant scale up batches and then commercialization, that is exhibit batches and commercialization, which you will submit to the regulatory agency. So during pilot plan also you can apply QBD, or you have already identified what are the critical material attributes and critical process parameters. Okay, so you have taken lab scale, you have studied in this range. Okay, but actually you have prepared that material for, that is lactose, for a particular attribute, that is particle size. You have studied in lab of 70 to 100 micrometers. Okay, but actually when you are developing your product, that is current product, you have taken 85 micrometers. And commercial process, this is design space. So this becomes your control, that is the parameter, and this is your design space, that is if you move anywhere in this space, that is if you take your particle size of lactose in this range, you will have a sufficient flowability and batch to batch consistency. That is the purpose of your control. Similarly, for all these parameters, this is with one attribute. Similarly, for other all attributes, you can define this design space, that is control strategy. You have to control the particle size within this limits, otherwise it will be having a other results or improper quality in your formulation. Similarly with process parameters, that is pre-roller compaction, blending, and roller compaction. So what are the equipments? So we have seen these are the parameters, which the variables which we have evaluated, and what is their range, what was actually used. Okay, and what can be the control space. Okay, so this are design control space and design space. And last parameter is product life cycle and continuous improvement. So after implementing all this throughout the product life cycle, the manufacturing process performance is monitored to ensure that it is working as anticipated to deliver the product with desired quality attributes. Okay, so it is a continuous process till your product is there in the market, that is product life cycle. Process stability and process capability will be measured and evaluated. If any unexpected process variability is detected, appropriate actions are taken to correct it, anticipate, and prevent future problems so that process remains in control. The additional knowledge gained through routine manufacturing can also be utilized for adjustment of process parameters, which can be a part of continuous improvement of drug product. So conclusion. So the goal of implementing QBD in formulation development is to reduce product variability and defects. So we have seen how severely we are evaluating every parameter of materials and process, how it is going to affect your CQAs. Okay, so if you are reducing the risk, so there are very less chances of variability and defects by enhancing product development and manufacturing efficiencies and post-approval change management. So you can move in your design space which you have created. So post-approval change management becomes easy. So it is achieved by beginning with the end in mind. So the result of development is a robust formulation and manufacturing process and establishing clinically relevant specifications. And finally, product and process capability is assessed and continually improved post approval during product life cycle management. So before I conclude, I just want to show uh, one more picture which is there. just 2 minutes. I wanted to show you one picture which gives you idea about QBD for novel formulations. Till then, if you have any questions, you can just type in chat box. Okay, so this is QBD for nano carriers, different from what we have seen that was tablets. Okay, so in that particle size, entrapment efficiency, zeta potential, and drug release, those are critical attributes. Then the polymers, the surfactants you are using, those becomes the critical material attributes. Homogenization, speed, time, temperature becomes process parameters, critical process parameters. Then using this, how they affect your particle size entrapment efficiency, you will perform the risk assessment using either flow charts or cause effect diagrams, create a design space. So these are the various, apart from factorial or fractional factorial design, you also have Plackett-Berman, Box-Penken, Taguchi, and response surface methodology. These are the different types of design of experiments which you can use to create a design space. Then control strategy, okay, and derive at your target profile. So first you have to define your target profile, then CQAs, and so this is a little bit different from what we do for tablets. That is the parameters. The elements remains the same, but the quality attributes and the CQAs, the material attributes, the critical process parameters change when you change your formulations. That is what I wanted to just show. And these are for the novel formulations which are there much nowadays research is done. So for those, these are the various of those parameters. Okay, so thank you. And this QBD is not only applicable to formulation development, you can also apply it for analytical method development as well as the active pharmaceutical ingredient. When you are manufacturing APIs, you are using different materials for synthesizing different process parameters like the catalyst or the temperatures and solvents. Even there, can apply this quality by design. Okay, so thank you. And if you have any questions, then it is open for discussion. Okay, so thank you, and we'll stop here. And I hope you will be able to apply this in your future. And I'd like to thank the SCIENTIA team and for giving me this opportunity, as well as principal ma'am and management for giving opportunity to to give a talk on this topic. Okay, thank you.
So good morning, everyone. It is such an honor for me to extend my gratitude to our distinguished speaker, Mrs. Meeta Jain ma'am, for giving us an excellent coverage on the topic QBD in formulation development. Ma'am has focused on various points like what are the difference between traditional and the QBD approach. QBD is basically a sound science approach, and how drug and excipients are susceptible to the risk and which could affect the quality. various life cycle approach to the quality by design, the advantages of the quality by design, what are the various elements and tools used in QBD, and also the steps to establish a process understanding. Our sincere thanks to Chairman Mr. Albert D'Souza, sir, for his encouragement, and thanks to all other dignitaries of the management. A special thanks to our principal, Dr. Sabita Toro, ma'am, for her immense support. I would also like to thank the coordinators for their guidance and support. We look forward to meet you all again in the upcoming sessions of SCIENTIA. The feedback link will be provided in your respective WhatsApp group. Kindly fill it. That will help us to improve our program. Thank you for being with us and have a good day.